Buenas tardes. Bienvenidos a este seminario de Frontiers in Genomics. Damos la bienvenida al Centro de Biotecnología Genómica de Reynosa, en Tamaulipas, del Instituto Politécnico Nacional, y a nuestros colegas de Mérida, de la UNAM. Y pueden seguir este chat en vivo en la dirección www.lsg.unam.mx/frontiers/live. Y es para mí un placer presentar al doctor Matt Berriman con su ponencia Sequencing the Tree of Life, perdón. Y voy a cambiar a inglés para hacer la presentación. Matt graduated in 1994 from the University of Manchester with a degree in biochemistry. He moved to the London School of Hygiene and Tropical Medicine at the University of Dundee to undertake the PhD with Professor Alan Fairlamb, biochemically characterizing a putative drug target for the malaria parasite Plasmodium falciparum. After being awarded a Wellcome Trust Traveling Prize Fellowship, Matt moved to the laboratory of Professor George Cross at Rockefeller Institute, University in New York to study trypanosome telomeres. In 2000, Matt joined the Sanger Institute as a senior computer biologist working on genomes of Plasmodium falciparum and Trypanosoma brucei. In 2003, he took over leadership of eukaryotic pathogen genome projects, and in 2008, he joined the Institute's faculty to establish a program on the genomics of neglected tropical disease parasites. Matt now leads the parasite genomics group, who studied the parasite that cause malaria and neglected tropical diseases. He uses a large-scale comparative and functional genomics approaches to discover and study genes associated with major biological traits of parasites or with potential for exploitation as targets for intervention. He's interested in the genes that underpin key differences between parasite lineages. These genes are often involved with pathogenesis processes and interactions between parasites and their hosts. He's particularly interested in helminths, including schistosomes, tapeworms, whipworms, threadworms, and hookworms, which cause some of the most neglected diseases and collectively infect more than 1 billion people. His team used comparative genomics as their main tool for sifting through the vast number of uncharacterized genes. Their largest study involving 81 species was published last year, providing a broad overview of most of the major roundworm and flatworms lineages that impact human health. Building on his background in comparative genomics, Matt was recently helped to establish a new scientific program at the Sanger Institute that is focused on the Tree of Life, which is the main topic of this talk. So with no further introduction, please, Matt, they are all yours.
Thank you, Alejandro, and thanks again for inviting me here to give this talk. I spent quite a lot of time yesterday talking to students about some of the research I've been doing over the last 20 years on parasite genomics, but I thought today I'd talk about a completely different area. So there's going to be no published work at all in this talk. This is going to be totally forward-looking about a new area of science that we're trying to develop at the Institute. So this is the Sanger Institute, where I'm from. It was actually set up as a center originally, so just with a single-minded purpose of joining the Human Genome Project effort. And we went on to contribute about a third, just over a third of the overall data in the Human Genome Project. Alongside that, a unit was set up to produce reference genomes for some of the most important pathogens of humans as well. And that's the unit I joined in 2000. The center has then grown over the years into a sort of fully fledged institute, really. This is it here, and also a bit of it over here. But it's on a campus with other institutes. So the European Bioinformatics Institute is there, plus some companies have joined as well. So it's a campus with genomics at its heart. But one thing that's kind of striking about Sanger is our purpose, our mission, if you like, has been to put genomics at the center of research aimed at focusing on the biology of humans and pathogens to improve human health. So we've had really very much a biomedicine agenda. And what I want to talk to you today is about another agenda, because actually Sanger has not really focused on quite a major part of biology. And that's ecology. So what I want to talk about today is a little bit about ecosystem services. So ecologists have referred to the way ecosystems provide for humans as ecosystem services for quite some time. You can divide it into many aspects, but really what we're interested in for the purpose of this talk is setting up a potential for intelligently looking after our ecosystems, but also mining them for things that are useful for us. Our relationship with ecosystem services is absolutely critical for the health of human societies. And this kind of agenda is more important than ever, given that we're facing absolutely catastrophic climate change, without question, and massive environmental degradation largely due to the increased urbanization and growth of human populations. So we have an urgent need to understand the structure and function of the communities of organisms that support us and all other life on Earth. With this in mind, an international consortium came together under the banner of the Earth Biogenome Project and published this announcement in PNAS 2 years ago, which they described as a moonshot for biology. And the goal of this project is actually to sequence all complex life on Earth. So they want to sequence every eukaryotic species on the planet. And the rationale for this is that firstly, it is now possible. It hasn't been possible in the past. The technology is ready that it's possible to sequence everything. And while we're trying to sequence everything, we'll no doubt discover new species. But also, the other thing that makes this moonshot highly relevant now is that we're facing record numbers of species becoming extinct. So unlike the early days of the Human Genome Project, here, the thing that we would potentially be trying to sequence is actually disappearing. So this project could allow a new foundation for biology, allowing us to drive solutions to preserve useful biodiversity and sustain human societies. So this is taken from the website of the Earth Biogenome Project, where they describe the benefits of undertaking a project like this. So the biome of Earth has had 3.5 billion years to evolve all kinds of novel solutions which could potentially be exploited for creating new fuels, the raw materials for biosynthesis, all kinds of starting points for mining new medicines and new ways of tackling emerging infectious diseases. So one could ask the question, how does this fit at an institute like Sanger that has really been so focused on biomedicine? Well, we have, but like I've already alluded to, we've worked on other things as well. So my group and others at the Institute have been working on important pathogens such as these worms, also malaria parasites, the mosquito vector of malaria parasites. Also major programs of biology at the Institute have looked at key model organisms such as zebrafish, mouse. Actually, the founders of Sanger worked on C. elegans, and of course, absolutely thousands and thousands of reference genomes for bacteria. So we do have a background in the comparative genomics of diverse taxa. And actually, we recently celebrated our 25th birthday. And as a celebration, we decided to sequence 25 species to celebrate these 25 years. And the way we picked these species was partly a driving force behind that was to try and engage the hearts and minds of the British public. So we came up with a list with a bunch of different rationale for selecting these species. Some of them were difficult to understand species, so otherwise known as cryptic species. Some were dangerous in that they're invasive and they're damaging our ecology. An example here is the New Zealand flatworm that's been imported into the UK and actually is a predator of earthworms. So that's very dangerous in terms of destroying our soil ecosystems. And there's invasive plants and so on. There's also species that are really on the watch list as floundering, others that are flourishing. So it'd be interesting to know the reasons why some organisms are struggling and some aren't. Then we have some that are simply iconic. So those things like the European red-breasted robin, again, just gets attracts the hearts and minds of the British public. And then there were a bunch of species that were put to a public vote. So the public actually voted to have these sequenced. Anyway, these have now been sequenced using long-read sequencing. So an important aspect of this project is we wanted them to be reference genomes. So they're sequenced once, and they serve as a reference for biology forever. And these are the various sequencing results, which maybe you can't see from where you're sat. But compared to genome projects of the past, we have really staggering assemblies now, with contig and scaffold N50s in the megabase range, many megabase range. Some of these now have assemblies where they're more or less as whole chromosomes. And here's these assemblies summarized. So in blue, we have the length of the assemblies. Remember, the human genome would be along here at 3 gigabases. So we have a range of assemblies from around about 100 or so megabases all the way up to, I think it's 9 gigabases here. And you can also see very large scaffold N50s. So this is a measure of the contiguity of those assemblies. Not clicking hard enough. So another factor in thinking about the relevance of us moving into this more ecologically focused research area is there's been a bit of a change in the attitude of our major funder. The Sanger Institute is largely funded by Wellcome, which is a global charity, and they now have a very prominent Our Planet, Our Health agenda, where they're really concerned in exploring the ways that that humankind is impacting the planet, but also could benefit from better exploiting the planet in more sustainable and intelligent ways. So with that in mind, we recently established the Darwin Tree of Life project with a bunch of partners from around the UK. Of course, we don't have a track record in biodiversity. We don't claim to have, but we've partnered with people that do, such as the Natural History Museum or the Royal Botanic Gardens at Kew, which maintain the Millennium Seed Collection, where they're trying to have at least one example of every plant on the planet. And there are others with fantastic track records in this area. The University of Oxford is partnered here with its field site called Whiteham Woods, which is an area outside Oxford, just of traditional woodland, but has been intensely surveyed for 80 years. It's perhaps the most ecologically surveyed bit of land on the planet. It's really quite extraordinary. There's barely a bird that flies through that woodland that hasn't been tagged or surveyed in some way. So the goals of this project, and it's in a pilot phase, we've been funded for the first 2 years, and a major part of the first 2 years is really kind of proving that we can do it. The whole project is not funded yet, but the partners have been funded to help us collect, and our goal is to generate chromosomally complete genome assemblies. Well, our overall goal over several years is to represent all 4,000 families of eukaryotes from Britain and Ireland. But in the first 2 years, we're actually going to be going for 2,000. And then later on, perhaps moving into a 10 or 12-year time frame, that's when we want to sequence all 60,000 species of Britain and Ireland. So why focus on Britain and Ireland? Particularly if you're an audience over here in Mexico, this seems like a fairly arbitrary geographic unit to study. Well, the first thing I'd like to say, that's not because I'm British. We actually have, there are several reasons to focus on this lump of land, and one of them actually is, in this first 2 years, it's an extremely logistic project. To prove that we can sequence a lot of species, we have to be able to get up and running fast. And if anyone in this audience has tried to move biological samples across international borders, they would feel some of our pain here, that actually the best thing to do, at least in this sort of startup phase, is go for species where you have a legal right to them. So we have kind of legal jurisdiction over this land. There's a whole load of politics about what we call this lump of land. It's really, for the purpose of this, it's an archipelago in the North Atlantic. It's also interesting because it's a bit of land that was stripped by the Ice Age. So it's almost been entirely recolonized in the last 8,000 years. So one could consider it as a model ecosystem for understanding climate change and recolonization. So here are some of the scientific goals of the project. Ultimately, I mean, these are pretty long-term goals. We'd like to study post-glacial ecological reconstruction. We'd like to use the sampling infrastructure to study some of the effects of climate change. There are species that we will be looking at that are actively monitored. So the same list of species have been surveyed, particularly around the intertidal zone around the British Isles for a number of years. We'd like to look at agro-ecosystem functioning, whether we can derive new feedstocks for biopharmaceutical mining, essentially, and also produce reference genomes for conservation genetics. So essentially the population genetics of species that are in decline. So the other thing to point out about this focus on a very geographically distinct group of species is it is somewhat representative of the global biota. So if we look in terms of the orders of the taxonomic orders, then actually those that the British taxa represent are actually more than half. We're going to be sequencing at the family level, and if we are able to sequence one species from every family, then we should have, at least at that level, covered 40% of the global families. And then ultimately our aim is to sequence all species. So according to current lists, that's about 60,000 species. And here's the list broken down a little bit more. It's clipped off on the right-hand side, but that doesn't really matter. You can see we have some quite low numbers of things like mammals, which is handy. And yeah, I mean, compared to Mexico, some of these numbers are really, really low, reflecting our very different climate. So what are we going to do over the next 2 years in detail? Like I said, our job now is to set up this kind of pipeline that we can go from any sample, like the garden snail here, and go all the way through sample receipt, barcoding it, storing stable specimens in museums, all the way through gene finding, and making the data available. So to do this, we need a network of partners. I've described them already. And there is the job of those partners. They're forming what we're calling genome acquisition labs. So they will organize taxon specialists to gather samples, create voucher specimens, and then when they've done that, send us a lump of tissue at the Genome Institute, where we will perform the high molecular weight DNA and RNA extractions and and sequence 2,000 single specimens. It's important for this that we're going to throw a bundle of different technologies at the problem, so that they have to be long reads sequenced. We ideally want close to chromosomes. There has to be long-range information such as Hi-C that I'll get onto, and optical mapping. The exact recipe we don't know yet. That's one of the things we want to learn. What's the best mixture to cost-effectively produce a complete genome? And on top of all of that, we've got to learn how to do it at scale, because ultimately to produce tens of thousands of species in that time scale, we need to be working to a point where you could potentially produce about 100 a week. This just summarizes those numbers. So that actually it's quite easy to flash big numbers up on a screen, but when you think about that, these really are huge compared to what we're currently capable of. So for our 25 genomes for 25 years, that took us about a year. So now we need to increase that output by orders of magnitude. There we go. So let's go through some of the things that we need to tackle. So getting a list of species is not trivial, actually. There are maintained lists, and actually they don't intersect that well. So our starting point is a list that's maintained by the Natural History Museum in the UK, but we're having to gather experts with a taxonomic focus to really pick those lists apart. For instance, a typical thing is you'll see a species on the list twice because it's been spelt in different ways, or the species name has changed. There's loads of species, such as the parasites, that aren't on any of the lists. They just haven't had that same kind of taxonomic focus. So there's a lot of work to do in getting those species lists right, and that won't be something we complete at the start of the project. It's something that will have to be ongoing while we're doing it. But we'll focus on some key areas initially where we can gather existing taxonomic experts to give us some tangible targets. And where possible, we want to store a voucher specimen in a permanent collection, such as the Natural History Museum. And of course, we're going to need to develop new extraction methods, and these extraction methods need to be tailored for the different sequencing platforms. And then we have to scale up all of these processes. They're all effectively kind of cottage industry processes at the moment, where in the past you'd hire a person to do one of those tasks. Ultimately, this project is going to require some standardization and automation. We're not there yet. Right. Oops. Right, so the first step on that pipeline is what we're calling the Taxon Task and Deliver groups. So we've established these genome acquisition labs, groups of collaborating institutes, and they, as I say, they need to validate species lists. You'll imagine, you can imagine this, it requires quite a lot of intensive debate and discussion, but we do have some species list emerging, and at the moment we're trying to pick these off in taxonomically focused groups, and part of that is then the research and development required around samples. We anticipate if you get a protocol that works very well for one insect, you might be then able to apply it to several insects rather than keep changing taxa every week. Then we need to, from those species lists, pick family representatives and make this list of the chosen few available to the public. But ultimately, we won't have the species available in people's freezers, so we'll have to go out and look at, but the taxonomic groups will have to go out and establish campaigns to actively seek species. So some of the criteria for the early priorities will actually involve some deep dives into key taxa, such as species that are iconic, important, or interesting. And I'll give you one example here. The butterflies of Britain, really a very small number of species. but it has an extremely engaged genetics community. So this is in the early days of a project such as this. It's very important that we have a scientific community on our side exploiting the data. Otherwise, there is no way we'll be able to maintain momentum. So we're picking off some of these sort of high-value groups early on. So let's have a look and see how we're doing on some of these butterflies. Just one more slide on the technology, just to emphasize again that we're having to use a range of different sequencing technologies. This includes long-read sequencing on PacBio or on Oxford Nanopore, scaffolding data from the 10x platform, from Hi-C and BioNano. So this is optical mapping. Again, we don't know the recipe yet. So in the years 1 and 2, we're having to do a bit of a pick and mix and try and work out what works. And so we anticipate the unit cost per species will be pretty high at the start of the project compared to when we can establish the most efficient method going forward. In addition to the sequencing methods, a lot is changing in terms of assembly methods. This is an area of really intensive work, and at the start of the project, just 1 or 2 years ago, it was incredibly computationally expensive to assemble a vertebrate genome. That is changing rapidly. Algorithmic design is really helping to pick apart complex diploid assemblies where you have bits of the genome where chromosome homologs overlap, whereas other bits of the genome where chromosome homologs don't align well because of heterozygosity. Assemblies need to tackle all of that complexity. So this is how we're doing with the Lepidoptera, the first 44 Lepidoptera, so the butterflies coming through the machines. On this plot, in blue were existing butterfly genome assemblies, and in red we have we have assemblies based on just raw sequencing data, and you can see already that's a giant leap forward just by having long read data. Also plotted on this graph is a assumed mean chromosome size, assuming they've all got 31 chromosomes, which is a hell of an assumption. That might approximate to something like the N50 if the genomes are perfect. But after some scaffolding, you can see, well, scaffolding the previous assemblies obviously has a big improvement anyway, but the ones in red sequenced as part of the Darwin project are now really approaching that theoretical limit of the assembly. And we know they're approaching that theoretical limit because some of the ones we've looked at, the assemblies are now looking really good. I'm going to walk through this one here for the red admiral butterfly. And a lot of credit here must go to folks at Pacific Biosciences, who, as well as making a lot of money out of selling us machines, have worked very closely on trying to help us optimize the protocols, and in fact have directly helped on some of the assembly work as well in the early days. So this is the first project where we've used the Pacific Biosciences Hi-Fi approach for sequencing. So this is the approach where instead of trying to push your read length to your fragment length to the absolute maximum, you go for a more modest fragment size, but sequence round the same molecule several times, and then on the machine, shrink that, you align those sub-reads for each molecule into a highly accurate consensus read, and do your assembly from those consensus reads. So it's an approach that, I have to say, PacBio have been pushing very hard. We were perhaps a little bit skeptical of it because we found the long templates just so useful, and we were a bit wary of excessive machine time as well. Anyway, so this is kind of an R&D project that they've really pushed quite hard, and the results have been quite spectacular. So these are the output stats from the smart cell. Nothing really interesting to report there. The assembly size has come out at the size you'd realistically expect. Not very many contigs. But if we look at the genome completeness, it's basically all there. If we look at the number of duplications, very low. So all the expected genes are there, present as single copies. Now, one of the key parts of the project is scaffolding using long-range information, but also quality controlling assemblies using long-range information. And we're finding at the moment high-C data is absolutely vital for these larger assemblies. Just a reminder of what we mean by high-C. So this is just taken from the Dovetail website. So high-C can start with endogenous chromatin or DNA that's where you reconstitute chromatin in vitro. But you cross-link proteins to trap proximity, chromosome proximity, digest away the intervening sequences, so you've got these protected chunks of DNA. After you've labeled the ends and then ligate those ends, the idea now is you want read pairs using short read sequencing that trap these informative sites. So they give you a map of bits of the genome that were near each other. So this is how the results look. So here we have an assembly and an assembly after some additional scaffolding work. And these are just interaction frequency maps based on the high-C data. Now, if we just drill into one of those locations, you can see before the additional scaffolding work, what you expect is everything to sit on a diagonal where there are far more interactions within a chromosome than between a chromosome. So whenever you see something like this, it means that this potential chromosome seems to be equally interacting with this potential chromosome, so they're most likely one continuous molecule. So with a limited amount of curation, you can go into the assembly and see if there's information to link those two things together. So having picked through these, it is quite a small number of potential errors, we then end up with a map of the genome that's almost completely error-free. And one of the nice things we spotted in it, it was the person at PacBio that spotted this, I should say, that some of our contigs from this assembly are utterly complete. So this really boring graph is actually a dotter plot of a contig against itself. And if we really zoom into that tiny dot there, we see this dotter plot. In a dotter plot, you've got a sequence against itself, and you do a dot every time the sequence is identical. And for a repeat, you end up with dots that are offset from each other. So this black shadowy area is actually a load of really, really tiny repeats going on for 11.3 kb, and they're actually sixmers. They're in fact the telomeric hexamer. And when we drill down into the other end and zoom in and zoom in and zoom in and zoom in, we end up with 17 kb of hexamers. So we've got one continuous sequence with no gaps in, starting with a telomere of 11 kb and ending in a telomere of 17 kb. So this is really very encouraging indeed. Another important experiment for us was to try and figure out an efficient way of not over-sequencing, because at the moment we tend to put one sample on one PacBio run, but as the yield of the machine improves, we don't want to over-sequence because we're going to just be burning through money. So here in this experiment, we're now looking at the amount of coverage for the red admiral butterfly versus the number of contigs you get out of it. And you can see as the sequencing goes up, the number of contigs goes down, but it plateaus out. So by the time you're over here, you're really wasting money. Likewise, if you look at the N50 stats, as you go from 10 to 15x, you're gaining a huge amount of assembly performance, but it does plateau out. And actually, as you get to really absurdly high coverage, you start introducing errors where scaffolds start falsely joining each other. So this has allowed us to provisionally set a kind of good rule of thumb, being 20 to 25x of these hi-fi consensus reads as our kind of recipe for what works on PacBio for now. And a similar experiment has been done on Mosquito down here. So the other way we're QCing our assemblies is looking at k-mer distributions. These slides are quite complicated to explain if you're not familiar to analyzing assembly k-mers. But what we have on the x-axis are k-mer multiplicity values based on short reads. So we take every k-mer in the short read and we count how many times we see it. So a homozygous site should be deeply covered. Let's say we're going for 100x coverage. You would expect that k-mer to be represented 100 times. If it's a heterozygous site, there'll be two alleles of it, so they won't be the same k-mer. Each one will be present at 50x. So we end up with these kind of haploid positions and diploid positions. Now, in the colors here represent the k-mer multiplicities derived from the assembly. So now in the haploid area, you'll notice we see a load of 0x k-mers. So these are k-mers that we know should be there from the short reads, but we can't find them in the assembly. But when the assembly is rerun using new tools like purge dupes to remove duplications and resolve the haplotypes, these missing k-mers are now found, so that black peak's gone, and this other peak here is filled in with k-mers that are present twice in the assembly. So the chromosomal homologs at that position have been separated out. So we're no longer creating these kind of Frankenstein haploid assemblies where alleles are effectively merged into representations that don't adequately represent either version. Right, just to say we are trying to optimize ONT sequencing as well. We've done a lot more work with PacBio, but ultimately we want to get ONT sequencing working as well. This is just to show some raw output that pretty much like any other ONT user, we're getting staggeringly long read lengths, really amazing, 40 or 60 kb, but our assembly pipelines haven't quite caught up with using the data in quite the same way as PacBio just yet. Right, so moving now down to the latter stages of this pipeline. I've mentioned assembly curation and a little bit, but I'm going to show another example of that, and then we're going to talk a little bit about data release. Ooh, what happened there? There we go. So I had to sneak in one parasite example because I'm a parasitologist. This is a very distant relative of the malaria parasite. It's a apicomplexan protozoan, and it's actually a genome project that I was involved in when it was published as a draft several years ago. And these are the stats from that published assembly. Wasn't bad. Over 50 megabases in a few thousand pieces. The chunks were quite big, and it had a decent busco completeness. But what we want is a perfect genome, actually, because it's a little bit of an aside, but the chromosomes had some pretty peculiar properties that we wanted to understand. There's a very strange banding of repeat content and a very strange banding of gene content that we'd like to drill into. Now, after sequencing by PacBio and doing some curation, we now end up with 21 scaffolds and much bigger N50s. And now let's have a look at that curation process. Oh yes, and of course the busco completeness is improved. So that, whoa. Uh-oh. Well, I can just click through. I don't know what happened there. Well, this is a spoiler, isn't it? Let's see if I can remember my place. Right. This slide, a high C plot, we've seen the one already. It actually doesn't look that bad, but because I've worked on apicomplexer for a long time, I'm kind of expecting something like 14 chromosomes, and this is way more than 20. And you can see all these hot spots all over the place where molecules are interacting with each other. And this led the curator to dig into the data some more, where they found a problem that's quite common to PacBio. And once you know it's there, you can obviously weed these kind of data out. When we think about how PacBio works, we have these templates with the dumbbell adapters, and we're monitoring the polymerase as it tracks along, goes around the dumbbell, and maybe goes around a few times. But occasionally we end up loading an individual smart cell on the machine with a template that hasn't been prepared properly, so it has an adapter at only one end. In this case, the newly sequenced molecule tracks along and then runs off the end because it doesn't have a dumbbell. And if we're unlucky, the sequence can fold back on itself. It doesn't need many palindromic sequences to do that. You know, if you've just got low complexity sequence, it will tend to fold back on itself, and then the polymerase will just track back using the original black sequence here as a template. So the read you get off the machine is just this black hairpin. It's a problem in a genome like Eimeria, where we have genuine palindromes all over the place, and they're biologically interesting, but then we have these machine-generated ones. But the trick to parse these out is they're very smart cell specific. So of your millions of smart cells you're looking at, you'll see the same palindrome only coming out of one smart cell and not the others. So now the curators spotted it, we're able to automate the process of not finding it again. So that's an example of where we need active curation to just kind of diagnose systematic problems, and then we can incrementally improve our informatics. Right, and here's the PacBio-based assembly. Looks pretty good, and then after curation, it's even better. Far fewer contigs, and you can see some complexities there being resolved. If we compare the curated version here to the old version, you can see it looked like a train wreck before, and now it's really it's in chromosomes. Right, so time's moving on. So after we've gone through curation, the next part of the project is annotation. Primary data release, so all of the reads are released automatically anyway, but our assemblies are going to be released automatically into the public archives, and very rapidly annotation is being performed by the ensemble group. So a lot of work has now been put in to try and close the time taken for annotation down to less than 1 month. So we have early and open data access, and then at the very end of all of this, a key thing we're now adding is that each genome is going to be turned in a semi-automated way into a publication through a collaboration with Wellcome Open Research. You may not have heard of this journal. It's like Faculty 1000 Research. It's a combination between an archive site and a traditional journal. Essentially, you deposit a manuscript there, and within 24 hours, it's live on the web. You suggest reviewers, and once 2 positive reviews have been received, then the paper is indexed and put in PubMed. But the whole time it's available and it's completely open access. And this is what, so far we have 3 that have made it all the way through. The 4th one is being worked on this week. And this is essentially what one of the papers will look like. There's very little extra to the paper other than this abstract. You know, that if you print it off, it's probably 2 pages long. There's some summary statistics, such as the high C plot and some assembly summaries, but it's just really a very simple data report. Importantly, it means that everyone who was involved in getting the sample and getting it to the point of data production are acknowledged on an author list. This is critically important for a project like this. We don't want people to be buried under consortial banners. If people have helped to produce the data, they need to have something on their CV. Otherwise, we can't rely on their help for future projects. And this is what our overall progress looks like as of last month. So the project has been running since October. Cheating on some of these numbers, because it does include the 25 Genomes project. So these ones that have made it through to publication are actually from the 25 Genomes project. But you can see we've got about 800 genome projects in the pipeline at the moment. I don't have any stats on typically how long it takes to go from here to here, but ultimately we need it to be something like a couple of weeks. So what challenges are we facing? Well, one of them is the fact that most organisms are not packaged in a conveniently large size. In fact, most of the biota on the planet, in terms of species, are very, very small. And at the start of the project, you really needed micrograms of DNA to prepare a decent library. But by intensely focusing on this area, there's been a bit of a breakthrough in that you can now produce a PacBio library from, well, in this paper, it was 100 nanograms. We've recently used the same approach on a single schistosome, which was 40 nanograms. So we're hoping that this can be pushed down to the sort of low nanogram range, ultimately. And the assembly that came out of this one mosquito, this is the assembly that was published several years ago, and this is the assembly that's come from the curated PacBio data from just a single mosquito, and you can see with its genome completeness score, it's a much better representation of the genome, in fact, clearly for a fraction of the time and effort and cost. This is just really an anecdote. Some species just turn out to be easy to sequence. We don't know why yet, but this funny little spider here, if you pull off 3 of its legs, you can make a DNA prep with exquisitely high molecular weight DNA. And that surprised us. We don't know why yet, but those are the sort of recipes we want to discover. But I guess the sort of elephant in the room, in a way, is still, there's an enormous amount of the biome that we're going to really, really struggle to sequence. Big furry things are pretty easy, but the single-celled biome is going to be really, really hard. So in terms of the Darwin Tree of Life project, we're not making very bold claims about how well we're going to get into that area, other than it's going to need some more focused R&D. We can't possibly work from fixed species lists in this area. A big part of the effort has actually got to go into discovering the species first. And the way that's going to happen, one of our partners, the Earlham Institute, are going to focus solely on this activity, and they're going to use a combination of single-cell sequencing with short reads plus long-range information captured from bulk samples to try and build up reference genomes of a sort, but unlikely to be of that sort of chromosome contiguity I showed earlier. Right. So one of the other things we're going to need in terms of building up a sensible tree of life, we can't just use local samples for everything. There's going to be lots of missing pieces. Of course, as scientific stories develop around these taxa, we can't just sample for purely logistic reasons. We need to fill in taxonomic gaps if we really want to start addressing questions framed around evolution. And that's where we're going to gradually have to tackle some of the text that's clipped off the screen here, where I'm pretty sure the Nagoya Protocol is mentioned. There's a huge area of regulatory affairs where, to collaborate globally on this kind of project, we do need to tackle the ability to move samples around the world. And ultimately, our goal, like I said earlier, is to not just stick to a couple of thousand species, but to ramp up from 2022 onwards to sequence the rest of the species list, which is really a huge level of acceleration that's going to have to happen. We're encouraged by the fact that there's been another mega project in biology. This project happens to be 300 years old. It's the Linnaeus system of classification. So biologists from across the globe have been classifying species in an incredible way for hundreds of years. If you added up the collective cost of this project by modern standards, it would undoubtedly be billions and billions of pounds. I mean, it's incredible that this reference resource is still used by all of us today. So in trying to actually produce a digital record for these species, we're building upon a project that's already been started and is in fact the oldest project in biology. I didn't want to just finish on that. I thought we had to at least consider the prospect of a Tree of Life project in Mexico. From my hotel room, I didn't do a very good job of surveying all of your biota. And if the numbers are incorrect, blame these sources. But I did find it quite marvelous that particularly things like our 15 reptiles, you've got 864 running around. In fact, I had a look at some of those reptiles today, and the number I looked at exceeded the total that we have in the UK. So it really is a reminder that, that although Britain and Ireland is kind of a tractable operational unit, it's really quite boring compared to what's out there. Oh, yeah, look at the butterflies. 59, you've got 1,800. Right. So that is... I have to finish on the acknowledgement slide. It's a real shame it's clipped off on the right, because there are some names I need to point out. Shane McCarthy runs the assembly team, and I've looted his slides like crazy. Mark Blackster leads our new Tree of Life project. Who else have I shamelessly stolen their slides? Kirsten Howe leads our curation team. And of course, we have loads of collaborators in academic labs and in industry and within the core facilities of our units, of our institute. And that's it. Thank you for listening.
You're looking at the Tree of Life, you're looking at individuals. How do you choose them? Because it's going to be the reference species. If, for instance, you look at, I just read something about rats in New York compared to Pittsburgh, I think. They're different. There's adaptation signals and stuff like that.
Yeah.
So if you're looking at conservation, that's going to be very important.
Yeah. No, you're absolutely right. And I think we have to kind of remember what's the purpose of a reference genome. So there's no way we can look at population diversity in one reference, so we shouldn't even try. What we have built into the project, which is relatively inexpensive, for those kind of canonical family representatives, we're hoping to sequence perhaps 20 individuals with Illumina technology, so we get some of the demographic information. But really, we're expecting these to be starting points where the research community can just take them and run with them.
Are you considering extinct species and museum samples?
Oh, you know, this is the biggest disappointment we've had. We really thought we'd struck gold when we signed up museums as collaborators, and so far we've not had good results from a single museum specimen, with the exception of the mosquitoes, where mosquito legs from these specimens that have been pinned to card in museums, these seem to give quite good assemblies. But everything else we've looked at, particularly if it's been in formaldehyde, is just sheared into 50 nucleotide pieces. It's disastrous.
Hi.
Hi.
So thank you very much for the talk. So we are actually sequencing a couple of birds and reptiles from Mexican biodiversity. So I was wondering about the different techniques and methodologies that you're using inside here. Can we, well, you already tried, I don't know how many genomes, and you know exactly.
Yeah.
Well, have an idea of, the pipeline, and what would be the best pipeline that works. So we are sequencing with PacBio and Hi-C.
Yep.
But we don't know the specificities of what techniques and at the level of bioinformatics to use next.
Well, contact me afterwards, because we do have some good trial and error kind of recipes now of swapping the order of different parts of the workflow in terms of things like scaffolding, and that can make quite a big difference. But yeah, we can dig into some of those details afterwards if you like. Yeah.
Thanks for the impressive talk.
Thank you.
Are you considering using any classical evidence like cytogenetics or so to really be sure on the number of chromosomes you should be getting? You didn't talk in that respect. Is this useful, you think, or not at all? Having the Hi-C, for example, for that.
This comes up quite a lot in internal discussions. We do have a cytogenetics core facility, and I think our current thinking is really that on some of the really large problematic genome assemblies, it's going to be worth that level of effort. But we just can't envisage at the moment having the throughput you would need, because so many, of course, they're so expensive, those kind of experiments, but they do give you critical information on particularly big and complex genomes, some of which we're avoiding. We're not going to, at the moment, we're not going to tackle the 670 gigabase amoeba genome, or some of the plant genomes are.
Because that would be my second question. Is somehow related. If you focus first at the family levels, you can have a really striking difference in genome sizes. And so again, any sort of previous evidence of genome sizes of some of these organisms to choose the perhaps smaller ones that may be easier to tackle and to get a better structure resolved?
History sort of tells us that we're not very good at predicting genome size at the moment. So I think we'll do the best we can based on current knowledge, but have to just kind of monitor the assemblies as they're being produced, because even in one sort of taxonomic group such as amphibia, where you might be expecting a 6 gigabase genome, there'll be outliers of 12 or 15 where it's important at this stage, I think, that we slam on the brakes and switch to a different species representative, because we can come back to those later as the technologies become cheaper.
I think there's no more questions. Oh, there's one here. Sorry.
Very different question, though. But I mean, thank you for this very nice and impressive project. But how do you manage to have, how many people are working on it, and how do you manage to organize the work so that by the end it fluxes nicely and...
Whoa, wow, that's a complicated question to answer. So it's an entire research program on the right. It currently comprises 3 faculty teams, so mine is one of them. So we're small research groups in this big department. And the department actually has some fairly professional kind of thinking from people we've recruited from industry who are excellent project and program managers. It became pretty clear in the first few months that was our Achilles heel, that really figuring out quite ruthlessly what you're not going to do now and you're going to leave till later is so important. Yeah. Yeah.
Well, now apparently there's no more questions, so I really want to thank Mac again and just thank you all for coming. Thanks. How can you pack by a protocol for very low interest?